Welcome student to the last part of the chapter 14, that is respiration in plants. Till now we have discussed the steps of respiration. First one was glycolysis, then it was Krebs cycle, ETS and how the ATP is formed. Now we will discuss the balance sheet of respiration. Net gain of ATP from each glucose molecule is calculated based on the following assumptions. Let's say all step in glycolysis, TCA and ETS occurs sequentially and orderly, which is a hypothetical condition, but we will assume that all these steps in glycolysis, TCA and ETS occur sequentially and orderly. The NADH which are synthesized in glycolysis is transferred into mitochondria and it will undergo oxidative phosphorylation. The third assumption is intermediates in the pathway are not used to synthesize other compounds. And the fourth one is only glucose is being respired. Other alternative substrates are not entered in the pathway at any stages. Such assumptions are not valid because all pathways work simultaneously and do not take place one after another. And the substrates that can enter the pathway and are withdrawn from it as and when necessary in the body. ATP, which is utilized as and when needed in the body as well. And finally, the enzyme reaction or the rates are controlled by multiple means inside our cell in the living organism. So such calculations are used to appreciate the efficiency of living system in extraction and storing of energy. Let's see the balance sheet of respiration. In glycolysis, the total amount of ATP synthesized is 2 and NADH are 2 molecules. In oxidative decarboxylation, that is Krebs cycle, 2 NADH are synthesized. In TCA, 6 molecule of NADH, 2 molecule of FADH and 2 molecule of GDP is formed. So if we convert according to ETS from glycolysis, 2 molecule of ATP and 6 molecule of ATP are formed. So total it is 8. From conversion of pyruvate to acetyl-CoA, 6 molecule of ATP are synthesized. And in entire TCA cycle, 18, 4 and 2. Total 24 molecules of ATPs are synthesized. And if we sum up, then we have from a single molecule of ATP, from a single molecule of glucose, we'll have 38 molecule of ATP. 2 ATPs molecules are spent for transporting 2 NADH molecules formed during glycolysis to the mitochondria. And hence the net gain is 36 molecule of ATP. Now let's compare fermentation and aerobic respiration. In fermentation, there is a partial breakdown of glucose. Net gain is only of 2 ATP and NADH is oxidized to NAD plus rather slowly. Whereas in aerobic respiration, glucose completely break down, that will release carbon dioxide and water. And the net gain of energy is 36 molecules of ATP. And finally, the NADH is oxidized to NAD plus very vigorously. Lastly, we will discuss about the amphibolic pathway. Glucose is a favored substrate for respiration. So all carbohydrates are first converted into glucose for respiration. But other substrates are also respired. Fats will break down into glycerol and fatty acids. Fatty acids are degraded to acetyl-CoA and it will enter into the Krebs cycle. Glycerol is converted into PGAL and it will enter into the pathway. On the other hand, proteins. Proteins are degraded by protease enzymes into amino acids and each amino acid after deamination will enter into the pathway at some stages in the Krebs cycle or as a pyruvate or acetyl-CoA. So here we can see that all the substrate like fatty acid, proteins and carbohydrates, their substrates or the path, they enter into the pathways. The respiratory pathway is generally considered as a catabolic pathway, but it involves both, that is anabolism and catabolism, that is synthesis of products and breakdown of products. So it is better, we can call it as amphibolic pathway. Example, fatty acid breakdown to acetyl-CoA before entering the respiratory pathway. But when the organism needs to synthesize fatty acid, the same acetyl-CoA will withdraw from the respiratory pathway. Similarly, during the breakdown and synthesis of protein, respiratory intermediates are involved. Now let's calculate the respiratory quotient or the respiratory ratio. Respiratory ratio, that is RQ, it's the ratio of the volume of carbon dioxide evolved during the course of respiration to the volume of oxygen consumed. So RQ depends upon the type of respiratory substrates. For carbohydrate, it is 1 because equal amount of carbon dioxide and equal amount of oxygen atoms are evolved and consumed. Here, 6 carbon dioxide, you can see on the screen that 6 oxygen molecules are involved in the respiration and 6 molecules of carbon dioxide is released. So the RQ, that is respiratory quotient of carbohydrate, is 1. Whereas the RQ of fatty acid, that is fat, is less than 1 because the amount of carbon dioxide released is less than the amount of oxygen molecule utilized. So it is less than 1. The RQ is given here. In living organism, the protein RQ is 0.9. In living organism, respiratory substances are often more than 1. So pure protein or fats are never used as a respiratory substrates. So it may convert into one or the other type of compounds, and carbohydrate is utilized as a respiratory substrates. Thank you for your attention.